right?
Fantastic. Yeah, that looks good. Thanks.
Okay. Thank you, Sam. So before I start my talk, I would like to acknowledge the Larrakia, the Daguman, Javan, the Wardaman people as the traditional owners of the Darwin and Katherine region, where we conducted our research, and pay our respect to their elders, past and present. So my talk will be an extension of the last talk that just Mary given about the CGMM and honeybee. In this talk, I will explain and discuss the experiment that we conducted and the results we got for the transmission of cucumber green mottled mosaic virus into watermelon via pollinating honeybees. So honeybees, European honeybees. We all know that honeybees are really important plant pollinators, and also they are frequent flower visitors. Bees are aid in seed production in flowering plants. In agriculture, the bee visits and the pollination increase the crop yield and the quality of the crop. In Australia, European honeybee is the main pollinator in the managed pollinator crops. So watermelon is one of the main crop that use managed pollination to increase the yield and the production in Australia. So because of that, and the watermelon is a fully pollinator dependent crop. Because of that, farmers place beehives around the farm to get the maximum production out of the watermelon. So it's recommended that at least keep the 3 to 4 honeybee hives per hectare to get the maximum production from watermelon. Bees not only important, they are also important in transmitting viral or the plant pathogen when they pollinate from those crops. So this viral transmission mostly a passive transmission where honeybees accidentally carry plant pathogen when they're visiting from an infected plant onto a healthy plant. This can happen via they just sticking onto their bee body and or bees carrying any infected pollen from an infected plant and then depositing back it onto a healthy plant. So study shows that these plant pathogens sometimes can replicate inside the bee body, hemolymph and in the bee bodies. And also some studies show that some of these plant pathogens can survive and be viable for longer time in the beehives that used for the crop pollination. So the particular the plant pathogen that I'm going to talk to you today is a cucumber green mottled mosaic virus. As you just heard from about the most of their epidemiology from the Mary's talk. So we found that this virus can survive inside the beehive. Not only survive, they can be viable for a long time inside some of the beehive products. So because the beehive that used to pollinate the infected properties can be the reservoir for this virus. If you use this beehive, there is a risk of transmission of this virus from the infected, the positive hives. So this is a concern in the watermelon industry as well as the managed pollination systems, because these positive beehives or any beehive these beehives are moved between properties and the crop to get the maximum pollination. Therefore, in this particular research, we wanted to test can honeybees transmit CGMME from these positive beehives onto healthy plants if they were used for pollination services. And also, we want to test can honeybees transmit CGMME from an infected plant onto a healthy plant if you get an infected plant on your property. So we conducted both laboratory and field trial to find answers for these two questions. So in the laboratory, we conducted trials to test first, can bees carry any of the CGMME material or the CGMME particles from one infected flower onto healthy flower, as well as we conducted a laboratory trial to find can bees carry any CGMME from positive beehives onto healthy flowers. So in the lab, we collected bees from a healthy beehive, and we placed that healthy bees on infected watermelon flower. We let bee to feed in that infected flower for some time. Once that bee finished feeding, we moved that bee onto a healthy watermelon flower. We let that bee to feed from the healthy flower again. Once it finished feeding, then we removed the bee from the vial, and that empty vial was placed on another healthy watermelon flower as a control. So we conducted series of these steps and the trials. After the end of those trials, we test all the bees we used and also all the healthy flowers and the controlled flowers we used in this trial for the CGMMV using molecular diagnostic techniques. In the lab trial, what we did, we collected bees from a positive beehive, and then we let that bee to feed on a healthy cucumber flowers for some time again. Once it finished, we remove the bee from the vial, and then we place the empty vial as a control again here. And at the end, we tested both bees and the healthy flowers and the control flowers for CGMMV. From the molecular test results, we found that half of the bees that were visited on to healthy flowers from our first lab trial were capable of carrying CGMMV from infected flower onto the healthy flower. And also, when we used the positive bees from the positive beehives, some of the bees were able to transmit CGMMV onto healthy flowers that they visit. However, the transmission is really low compared to the transmission of virus from infected flower in their transmission from positive hive onto healthy flower. So at the end of these laboratory trials, we found that yes, bees can carry CGMMV when they're visiting from both infected flower and from the positive beehive. So here I got a small video to show you how the bee behave inside a vial when they're allowed to feed from the flowers. So after the lab trial, we realized that yes, bees can carry virus, but we want to test these scenarios. Can they still carry virus in the field, and could that lead to an infection in the field? So we want to test again, can bees carry virus from infected plant to healthy plant? And because of that virus transmission, can that plant get infected? And second one, can bees transmit any virus from positive hive onto healthy plants? And again, could that viral transmission lead to a new infection in the field? So in the first field trial, we placed CGMA positive watermelon plants at flowering stage in the field site. And in between those positive watermelon plants, we placed healthy watermelon plants at flowering stage. And also we placed watermelon plants with no flower as a control for this field trial. We used 2 CGMA free beehives for this field trial. And then we allowed bees to pollinate both infected and healthy plants for 5 consecutive days. So we expose our experimental plants and control plants for bees only in the morning hours from 8 a.m. to 12 noon, because that's the most active period of the bees on the watermelon flowers. So in the afternoon, we covered these experimental plants and control plants, avoid further bee visit in the afternoon. So once experiment plants had the 5 days worth of bee visits, we placed this plant in a biosecure glass house and further grown for another 8 weeks. During that 8 weeks, we monitored these plants for any CGMMV symptoms, and also we remove any insect pests that we observed from those plants. So at the end of 8 weeks, we tested our experimental and healthy plants for CGMMV. In the second field trial, we followed the same setup, but instead of having infected watermelon plants, we only had healthy watermelon plants at flowering stage. And also we placed some watermelon plants with no flower as a control for this one. These plants were exposed to a CGMMV positive beehive, which we bought from an infected property. And then we allow these bees to pollinate our experimental plants. So in this particular field trial, we had 4 different plant treatments. In the first plant treatment, we had healthy watermelon plants and the control plant exposed only for 1 day. Within 12 hours of the hive being exposed to CGMA positive plants. In the second treatment, we had another set of plants we exposed for 5 consecutive days after 36 hours of the hive exposed to CGMA positive plants. After we finished first 2 field treatments, or finished first 2 plant treatments, we removed the positive beehive from the field site, and then we let it resting for 2 weeks in a place where there is no known CGMMV. After it's finished its resting at the site for 2 weeks, we brought the hive back onto the field site, and we exposed another set of plants for 1 day 1 day for the bees, and then they also expose another set of plant for another 5 consecutive days after it has been rested in a resting site. So after each treatment, we place these plants in the biosecurity glasshouse again. We monitor those plants for 8 weeks for symptoms, or we remove any insect pests that we observe from those plants. And at the end, we tested this plant for CGMMV again. So from the first field trial, what we observed, more than 80% of the plants we used in the first field trial were tested positive for CGMMV. From these plants, we also observed some the CGMMV symptoms on their leaves. So how about from our second field trial? From the second field trial, we detect CGMA positive plants only from our first treatment. We didn't detect any CGMA positive plant from our other 3 treatments that we had in the second field trial. So from these 2 field trials, what we can confirm, yes, these can be vector and transmit the virus in the field, and also that viral transmission can lead to a new infection. However, again, the CGMA transmission is really low from positive beehives compared to the CGMA transmission from infected plants. So just to summarize all our results, at the end of this lab trial and the field trial, now we can confirm that yes, honeybees can be vector for CGMMV. However, transmission is really low from CGMA positive beehives compared to the transmission from infected plants in the field and in the lab. So this suggests us to look for some mitigation risk associated with CGMMV be in this system. So we would recommend to remove an early detection and removal of CGMMV infected plant from your properties to reduce the further spread of this virus via pollinating honeybees. And also we recommend to have a resting for the beehives that were used for infected property for at least 1 month before it's introduced into a new cucurbit properties to stop or mitigate the risk associated with the CGMMV transmission from positive beehives. So I would like to thank all the growers and apiarists who contributed to this research, and also the Horticulture Innovation and also Australian government for funding this research. So if you want to know or read more of our research outcomes, fact sheets, reports, and presentation that we given from our whole project, I would suggest you to just check in the Horticulture Innovation website or just shoot an email for us. We'd be happy to give you more details of the fact sheets about this project. So that's the end of the talk. And then I would like to thank you all for listening.
Thanks very much, Darsh. That was great, and great to have you straight after Mary as well. Has anyone got any questions for Darsh? We've got time for a couple.
Hi.
Darsh, can you stop sharing if that's all right?
That's all right.
I can't see the participants. Was someone asking a question?
Yeah.
Oh, Park, yes.
Yeah. I'm wondering, would you monitoring those virus positive hives frequently, or maybe and because I'm wondering maybe just like randomly sampling those workers, and
so you mean for the field trial one, right?
Yeah, because I'm thinking because you shows that a low transmission rate from the virus positive hive to the plants. I'm thinking maybe they carry the relative low titer of the virus to the plant, right? Is that possible?
Yes, actually for the field trial too, that we use the positive beehive, we actually sampled bees, the foraging bees from the landing board every day throughout our experiment, and we tested all those bees for CGMMV. I haven't presented it here, but interesting what we found, the CT values from those bees we tested over the time was increasing. That means their viral load was decreasing over the time. Yes, we observed that one from our positive bees.
Yeah, very interesting. Thanks.
Thanks, Hark. Anyone else got a question? We've got time for one more. All right. Well, then we'll move on. Just to let you know as well, at the very end of the next symposium, we have got a panel discussion. So if the speakers are available, then